In this class, we'll try to learn about pregnancy and parturition. The learning objectives will be students should realize the importance of learning physiology of pregnancy, understand physiology of pregnancy at different stages starting from fertilization to fetal maturation, list the placental hormones and mention their functions, list causes of female fertility, outline the concept, indications, steps of in vitro fertilization, describe various systemic maternal changes in pregnancy, explain uh, briefly the mechanism of parturition, and elucidate the role of oxytocin in parturition reflex. The transport of egg following ovulation, the oocyte is extruded onto the surface of ovary into the abdominal cavity. The fimbriae located at the end of the fallopian tube actively picks up the oocyte. The fimbriae contain smooth muscle and are lined with ciliated epithelium. Immediately after ovulation, smooth muscle of the fimbriae contracts, which makes the fimbrial end of the tube to migrate close to the oocyte. The cilia of fimbriae beat the waves towards the interior of the duct. These ciliary motions sweep the egg into the fallopian tube as egg emerges from the follicle into the ovarian surface. Inside the fallopian tube, egg movement is driven by fallopian tube cilia, which normally is a slow process. Therefore, the egg takes about 4 days to reach the uterus. Hence, the fertilization to occur, it must naturally happen in the distal portion of the fallopian tube, that is within a day of ovulation, because of the short viability of the unfertilized egg. Transport of the sperm. Transport of the sperm in the cervix. Passage of sperm through the cervix is favored by the nature of cervical mucus. The sperm transport is favored by estrogen induced changes in consistency and orientation of the mucus. The estrogen makes the mucus thin and elastic. And also, under the influence of estrogen, cervical mucus is oriented in parallel that favors the transport of sperm. Thus, sperms can easily swim through the estrogen dependent cervical mucus. The transport of the sperm through the entire length of the uterus and then into the fallopian tube occurs due to the sperm's own motility and uterine contractions. In fallopian tube, sperm motility is facilitated by three factors. One is ciliary movement of the tube, second one is peristaltic activity of the tube, and then the flow of fluid in the tube. Though several millions of sperms are deposited in the vagina, only about 50 to 100 reach the fallopian tube. This is the major reason that there must be many millions of motile sperms in the seminal fluid for fertilization to occur. Thus, a decreased count and motility of the sperm lead to infertility in males. Changes in sperm in female genital tract, sperms undergo changes in the female genital tract that makes them able to fertilize the egg. These changes are collectively called as capacitation, which occurs after sperm spend one or several hours in the female genital tract. The capacitation includes two major changes in the sperms. The wave-like beating of the tail of the sperm is replaced by whip-like movements that propel the sperm towards the strong lurches. Alteration in the plasma membrane, that is modification of the surface proteins of the sperm makes them capable of fusing with the surface of the egg. Freshly ejaculated sperm cannot immediately penetrate the ovum. Only capacitated sperm is capable of doing so. So here uh, there are different stages of fertilization of implantation. First is ovulation. Second one is the picking of the egg of the oocyte by the fimbriae. Third one is fertilization with exclusion of the second polar body. And fourth one is zygote formation. So then two cell stage, then four cell stage. Okay, so then the formation of the early morula, late morula, so then blastocyst. Okay, so then this is the stage of early implantation. Fertilization is the process of union of sperm with egg that results in zygote formation. Normally, fertilization occurs in the ampullary portion of the fallopian tube. Fertilization takes place in four steps: fusion of sperm with the egg, then acrosome reaction, sperm penetration, polysomal polyspermy block, and formation of the zygote. So, fusion of sperm with egg. It begins with the fusion of a sperm and egg. Many sperms move between the columns, uh, cumulus cells, that is granular cells still surrounding the egg, to bind to the zona pellucida of the egg. The zona pellucida contains glycoprotein that serve as a receptor for sperm surface proteins. The sperm contains many such proteins and therefore bind simultaneously to many receptors on the zona pellucida. Acrosome reaction. The binding of a sperm to zona pellucida triggers acrosome reaction in the bound sperm, which is required for sperm penetration. The steps of acrosome reaction and penetration of the sperm are as follows. First, there will be change in the membrane of the sperm head, that is, redistribution of the constituents of the plasma membrane of the sperm head at the site of attachment results in alteration in the membrane. Then increase in the fluidity of the membrane of the sperm head, then increase calcium permeability of the sperm membrane, digestion of the part of zona pellucida, that is the membrane bound acrosomal enzymes in the sperm head exposed to the zona pellucida. So these are proteolytic enzymes that include acrosin, a trypsin like protease. They digest the portion of zona pellucida at the site of binding. Then penetration of sperm through perivitelline space. As the zona pellucida gives way, sperm advances through coating using its propulsive force of the tail. So then sperm penetrates the perivitelline space, that is the space surrounding the egg membrane, to reach the egg plasma membrane. Fusion of sperm head with the egg membrane. So the sperm head fuses with egg membrane, so which is facilitated by sperm surface protein called as fertilin. Then sperm slowly enters into the egg cytoplasm, that is ooplasm. Only one sperm penetrates the egg. So this is because as soon as the sperm penetration occurs, immediately a series of change occur in the egg that prevent the entry of additional sperm. So this is called as polyspermy block. There are two mechanisms of polyspermy block. The initial fusion of the sperm with the egg plasma membrane triggers a reaction that changes the membrane potential of the egg. So the change in the egg membrane potential prevents additional sperm binding. Second one, secretory vesicles that are located in the peripheral part of the egg cytoplasm release their contents into perivitelline space, a narrow space between egg plasma membrane and zona pellucida, by exocytosis. These vesicular contents enzymes that enter the zona pellucida and cause inactivation of the sperm binding sites on zona pellucida and hardening of the entire zona pellucida. So thus binding of additional sperm to the zona pellucida is prevented. Zygote formation. The sperm penetration of the egg is called as fertilization. The fertilized egg completes its second meiotic division in the next few hours. As a result of second meiotic division, the second polar body is formed, which is excluded from the ooplasm. The remaining haploid nucleus containing 23 chromosome is transformed into the female pronucleus in 2 to 3 hours. Two sets of chromosomes, that is 23 from egg and 23 from sperm, each surrounded by distinct membrane known as pronuclei, the male pronucleus and the female pronucleus, migrate to the center of the cell by contraction of the microfilaments and microtubules. In the meantime, DNA of the chromosome in both pronuclei replicated and pronuclei fuse. Then the pronuclear membranes break down. Now the cell is ready to undergo mitotic division. So this completes the process of fertilization, and the fertilized egg is called as zygote. The fertilized egg remains in the fallopian tube for 3 to 4 days, while in the fallopian tube, a number of mitotic cell divisions takes place. So this process of mitotic cell division is known as cleavage. The first cell division takes place about 24 hours to produce two unequal size cells. The structure is called as blastomere. Then each cell divides into form a 4 cell conceptus, 8 cell conceptus and morula, where 16 to 32 cells are formed in 42, 72 and 96 hours respectively. Twins are formed if the dividing cells grow into two independent cell masses. After fertilization, the conceptus slowly moves from ampulla to the fallopian tube into the uterus. Till the morula stage, conceptus is in the fallopian tube. Until implantation in the uterus, the conceptus is enclosed in the zona pellucida. The intact zona pellucida has three functions. That is, it prevents adhesion of the conceptus to the inner wall of the fallopian tube. It protects the conceptus from mechanical damage while being transported from ampulla of the tube into the uterus. It also prevents the early immunological rejection of the conceptus. Blastocyst formation and implantation. The morula enters the uterus about 4 days of fertilization. After reaching the uterus, the conceptus floats in the free intrauterine fluid for approximately 3 days, during which it further undergoes cell divisions. Now the conceptus is known as blastocyst, the stage at which the cells have lost their totipotentiality and begin to differentiate. The blastocyst derives nourishment from the uterine fluid. The blastocyst consists of three components, that is trophoblast. An outer layer of cells of blastocyst is called as trophoblast. The trophoblast consists of extraembryonic ectodermal cells present on embryonic membrane. It helps in implantation of the conceptus and contributes to placenta formation. Trophoblast and its membrane secrete human chorionic gonadotropin, that is HCG. Embryoblast. An inner cell mass is called as embryoblast that gives rise to the fetus. Then blastocele, a central fluid filled cavity of blastocyte is called as blastocele. It forms amniotic cavity. In subsequent development, the inner cell mass gives rise to the fetus. The trophoblast surrounds the embryo or fetus throughout its development and is involved in its nutrition and as well as in the secretion of several hormones. Implantation and early development. Implantation is the attachment of the blastocyst to the endometrial surface of the uterine wall. It occurs about 7 to 8 days after fertilization. Implantation occurs in estrogen and progesterone primed uterus. By the time of zygote develops into the blastocyst, the menstrual cycle reaches about 21st of the normal menstrual cycle and 7th post-ovulatory day, during which the endometrium is being prepared by progesterone secreted from the corpus luteum. During this period, embedding of the blastocyst into the endometrium, implantation begins. The progesterone prepares the endometrium for the implantation of blastocyst. The trophoblast cells are very sticky, especially at the region of that overlie the inner cell mass, and this part of blastocyst adhere to the uterine wall for the implantation. The implantation outside the uterus leads to ectopic pregnancy. So these are the different stages of blastocyst, that is 2-staged, 4-staged, and this is called as morula, and this is called as blastocyst stage with the trophoblast, blastocyst and embryoblast. So this is the part of the uterus where it is going to be implanted, and it is called as decidua, which contains the compact spongy and the basal and the muscular part. So this is the different stages of formation of the placenta. Initially there will be formation of the amniotic cavity, And so then the formation of the amnion and chorion, that is around 10th week, and then there will be a formation of the proper placenta at around 12th week. Implantation is completed in about 10 to 12 days after ovulation. The trophoblast cells become cytotropoblasts, that is large polyhedral cells that are surrounded by syncytial trophoblasts. So these are the cells with no boundaries. The blood vessels in the endometrium dilate and form lacunae. Chorionic villi, the functional units of placenta, begin to form at about 12th day from the fertilization. Placenta is a combination of interdigitating fetal and maternal tissue that serves as an organ of exchange between mother and fetus during pregnancy. The fetal part of the placenta is formed by the outermost layer of trophoblast cells. The chorion, and the maternal part is formed by the endometrium underlying the chorion. Chorionic villi, that is finger-like projection, originate from the trophoblast cell and extend from the chorion into the endometrium. The chorionic villi contain rich network of capillaries. Each villus is surrounded by pool of maternal blood called as placental sinus supplied by the maternal arterioles. Arrangement of the placental circulation. The maternal blood flows into the placental sinuses via uterine artery and come out via uterine veins. Simultaneously, the blood flows from fetus into the capillaries of the chorionic villi via umbilical arteries and come out of villi back to the fetus via umbilical vein. All these umbilical vessels are contained in the umbilical cord, a long rope-like structure that connects the fetus to the placenta. Formation of amnion. Once placenta is well formed, the fetal heart begins to pump the blood at about 5th week of gestation. A layer of epithelial cells in the villi and a layer of endothelial cells in the fetal capillaries separate the maternal and fetal blood. Exchange of materials between maternal and fetal blood occurs between these layers. A space is formed between the inner cell mass and the chorion is called as amniotic cavity. The cavity enlarges and it's slowly lined by the epithelial cell layer derived from the inner cell mass called amnion or amniotic sac. Amnion finally fuses with the chorion to form single combined membrane that surrounds the fetus. The fluid in the amniotic cavity is the amniotic fluid that buffers the mechanical stress and temperature variations. The fetus floats in the amniotic cavity. The fetus is attached to the placenta by umbilical cord. Eventually, it is the only amniotic sac that separates the fetus from the uterine lumen. So this is how the amnion is formed, and this is how the chorion is formed. So fetoplacental unit. Some of the substances they are not produced by the placenta, and some of the substances or the hormones they cannot be produced by the fetus. So there will be change of substances which helps in the formation of these substances. So placenta produces the cholesterol and progesterone, and progesterone is transferred from the placenta to the fetal uh, blood where they get converted into cortisol and corticosterone and androgens, okay, so which are required for the fetus. Okay. And sometimes the placenta will not be able to produce estradiol and estriol, okay, so which are produced in the uh, fetal adrenal tissue utilizing the pregnenolone, which diffuse to the adrenal cortex. Okay, so then with the help of dihydroepiandrosterone, the placental tissue produces estradiol and estriol. Okay, so, and this the interaction between placenta and the fetal adrenal gland for the steroidogenesis is called as fetoplacental unit. Female infertility. Infertility is defined as the inability of a couple to achieve conception after 1 year of unprotected sexual intercourse. In females, infertility may be due to the following causes: ovarian factors, anovulation, that is, absence of release of ovum from the ovary, causes infertility. Peritoneal factors, pelvic adhesion or pelvic peritonitis prevents fallopian tube to pick up the ovum from the pelvic cavity. Tubal factors, partial or complete bilateral tubal obstruction resulting from inactive or ineffective salpingitis, infective or non-infective salpingitis prevents the fertilization to occur. Uterine factors like endometritis, intrauterine adhesions, or anatomical defects can prevent implantation of the fertilized ovum. Cervical factors like thick cervical mucus or presence of sperm antibodies in the mucus makes the sperm impenetrable. Vaginal factors like infections of the vagina or anatomical defects in the vagina prevent sperm to reach the cervix. In vitro fertilization and embryo transfer. So this is the field of reproductive medicine which has changed forever with the birth of Louis Brown in 1978 by in vitro fertilization and embryo transfer. Patrick Steptoe and Robert Edwards of England are remembered for their revolutionary work. The past decade has witnessed two more dramatic changes in the technique protocol of IVF-ET. One such change was from natural cycle to superovulation protocol, and the other one was replacement of laparoscopy by vaginal sonography for ovum retrieval. So, how the patients are selected? The patient age should be less than 35 years. There should be presence of ovarian reserve at day 3. The serum FSH level should be less than 10 units per liter. Then husbands should have a normal seminogram. The couple must be screened for HIV and hepatitis. The normal uterine cavity is evaluated by hysteroscopy and sonohysterography. So then these are the principle of the artificial reproductive technique. So initially there will be down regulation of using the GnRH agonist. Then there will be controlled ovarian stimulation. There will be monitoring follicular growth. Then there will be oocyte retrieval. There will be fertilization in vitro, transfer of gametes of embryos, then luteal support with the progesterone. Placental hormones, human chorionic gonadotropin. The human chorionic gonadotropin is the gonadotropin released from the syncytiotrophoblasts of placenta. The syncytiotrophoblasts cells start producing HCG between 6 to 8 days after fertilization. It's a glycoprotein hormone containing galactose and hexosamine. It is made up of two subunits, that is alpha and beta. The subunit contains 92 amino acids and has a molecular weight of 18,000, whereas beta subunit contains 145 amino acids and has a molecular weight of 28,000. The alpha subunit of HCG is similar to that of alpha subunit of LH, FSH and TSH. Human choreographic, the HCG secretion is more in the early pregnancy. It appears in the maternal plasma as early as a 6th day after fertilization. Its concentration increases rapidly to reach peak about 10 to 12 weeks of gestation, and then concentration is about 5 milligrams per ml. Then the concentration decreases up to 75% at about 25th week and remains that even till the term. And associated with the decline in HCG secretion, the placenta begins to secrete estrogen and progesterone. Functions. The human chorionic gonadotropin binds to LH receptors in the corpus luteum. Therefore, it has mainly luteinizing and luteotropic activities. It has very less FSH activity. It stimulates progesterone production from corpus luteum. It plays a critical role in placental steroidogenesis. It also helps in the sexual differentiation in male fetus. It stimulates the emitting centers in the brain. So therefore, morning sickness, that is sense of nausea and vomiting in the morning, is an early feature of pregnancy. Morning sickness is common in first pregnancy and usually disappear after the first trimester. HCG is detected in plasma as early as 6 days after conception. Therefore, demonstrating HCG in plasma by radioimmunoassay is a pregnancy diagnostic test. HCG appears in urine from 14th day of conception onward. Detection of HCG in urine is easier as blood collection is avoided. Therefore, HCG is not specific for pregnancy as it is secreted from many gastrointestinal and chorionic tumors. It is also secreted in some amount from the fetal kidney and liver. Human chorionic cementotrophin. The human chorionic cementotrophin is also called as human chorionic somatomamotrophin or human placental lactogen or chorionic growth hormone. It is secreted from the syncytial trophoblast of the placenta. So it is similar to the human growth hormone. HCS, GH, and prolactin are found from the common precursor hormone. So it contains 191 amino acids and has a molecular weight of 22,000. Secretion of HCS starts from third week of pregnancy and slowly rises till term. The peak concentration in plasma is achieved at term and it is around 15 milligrams per ml. Human chorionic somatotrophin has a function similar to the growth hormone and prolactin. It promotes growth of mammary gland and stimulates the production of the milk. Therefore, it is called as human placental lactogen. However, HCG is less potent in milk production than prolactin secreted from the anterior pituitary. It stimulates the somatic growth of the fetus. Therefore, it is also called as growth hormone of pregnancy. It causes maternal lipolysis and releases free fatty acids from fat tissues. It decreases glucose utilization in mother. It induces maternal insulin resistance and glucose intolerance. It alters fuel availability for the fetus by antagonizing the maternal glucose consumption and enhancing fat mobilization. So this ensures adequate supply of fuel to the tissues. So clinical significance is the quantity of HCS secreted is proportional to the size of placenta. So therefore, a low HCS level in the third trimester is the indicator of placental insufficiency. The other hormones like relaxin secreted from the placenta causes uterine relaxation in the early part of the pregnancy, like progesterone, to facilitate implantation and prevent expulsion of the fetus. Its concentration in plasma is highest, that is 1 nanograms per ml in the first trimester of the pregnancy. Towards the term, it causes relaxation of the pubic symphysis and pelvic ligaments to facilitate delivery of the fetus. Progesterone causes myometrial quiescence and promotes the continuation of the pregnancy. Estrogen secretion increases slowly in the third trimester and reaches peak at term. The major estrogen secreted in pregnancy is estriol, which normally is secreted in very less amount from ovary of non-pregnant woman. Estrogen facilitates parturition.